investigadores de diversos países, de diversas universidades, también chilenos, que estamos aquí generalmente a las 2 hasta las 3:20, que lo esperamos, y que hoy tendremos como presentadora a la doctora Karmiloff-Smith, y la persona que lo introducirá será Marcela Tenorio, que está acá en la persona, y desde el micrófono para que introduzca. Si no, todavía más fuerte.
Gracias, hola a todos, buenas tardes. Es para mí un honor ser la persona que presenta a la profesora Karmiloff-Smith. Annette es licenciada en psicología y doctora en psicología genética experimental en la Universidad de Ginebra. Perteneció al Centro Experimental de Investigación dirigido por Jean Piaget, quien fue su tutor doctoral. Tiene una trayectoria de trabajo con él y es considerada una de las discípulas vivas más relevantes y más importantes en el estudio y en las contribuciones a la psicología del desarrollo en la visión contemporánea que tenemos de ella en este momento. Annette trabajó por varios años en la Universidad en UCL. Ahora pertenece al Centro para la Investigación del Cerebro y el Desarrollo Cognitivo en Birkbeck University en Londres. Ha tenido una amplia trayectoria investigando en síndromes genéticos, en general en desarrollo típico. Trabajó por muchos años en síndrome de Williams y ahora viene con todas las novedades de la investigación que está sucediendo en Londres en este momento en síndrome de Down. En Londres se han formado un consorcio que es Downs. Es un consorcio multidisciplinario, enorme, multinivel. Ella les va a contar muchos más detalles de los que yo podría contarles, que está adelantando tal vez por primera vez investigación en tan distintos frentes para tratar de comprender este síndrome específicamente. Muchas gracias, Annette, thank you very much por estar aquí bueno, ella les va a contar su experiencia en español.
I'm really sorry, I can't speak to you in Spanish. Can everyone hear me, or do I need a microphone?
Microphone, please.
It's not working. I think
Is it working? Hello, it works? No. Okay, can you hear me now?
Yeah.
Yes? Good, okay. I'm a little bit far from my computer. So, I decided today, because it's a doctoral seminar, that I would tell you about my latest research, because I'm really excited about it. But on Tuesday, I will talk more generally about typical and atypical development. But today, I just want to concentrate on the new work. Okay. So, I think it's quite surprising that we're going to be studying babies, so really small children, when we want to understand dementia in adults or Alzheimer's disease. Everyone would say, well, you know, this is an adult disease. Surely you want to look at adults. Why study babies? And I'm going to tell you why. So first is a very general thing that I have argued throughout my career, and that is that the only way you can understand anything in terms of human psychology is to study it developmentally. I think if we really only by looking at the roots of development do we really understand how things develop over time and become the end product. There's something wrong with the microphone? Well, I'll shout for a moment and then just go on, and then maybe it'll be too loud suddenly. So, studying the origins in infancy, I think, is a very, very important aspect of developmental psychology to understand the adult end state. But development isn't just about studying children, it's about studying change over time. And that's a really important point. So you can study, I think I'm getting louder, you can study adults developmentally. And there are people now looking at healthy aging and the changes in the brain and cognition that occur over adult developmental time. Oh, it's getting too loud. Okay. So anyway, so the first point is study things developmentally. The second point that I want to mention, I think Macintosh to things change all my layout, doesn't it? There's a gene, so everyone knows Down syndrome has an extra copy of chromosome 21. And interestingly, one of the genes on chromosome 21, the APP gene, which is implicated in the amyloid beta plaques in dementia, is actually on chromosome 21. So from conception onwards, throughout fetal development, throughout postnatal development, this gene is getting overexpressed. So there are clues there to the fact that we might discover in babies some indications, if you like, of what causes adult dementia. And we know now that the brain pathology starts really early. So people who are making, you can't really see this very well, the colors have unfortunately changed. People who are making stem cells from hair follicles and from other human tissue have been able to show that in babies with Down syndrome, you already get the implications that the APP amyloid, beta amyloid plaques are forming already. So very, very early, you're getting the initial state of what will become in adulthood dementia. There's not only the APP gene on chromosome 21 though, there are other genes such as chromosome 19 where the APOE gene is associated also with dementia. And this gene has two allelic differences, one the 4, which is a risk factor, and the other the 2, which is a protective factor. So again, we're going to be looking at children with Down syndrome to see how these other genes on other chromosomes also contribute to the outcome. So risk and protective factors are two things we're going to be very sensitive to in this new study. And of course, all of these different genes interact. So there's not just a single gene and its output. Genes turn on, if they're transcription factors, they turn on other genes, they turn off other genes, they interact with them, they cause their expression, etc. So it's a very important and a very, I think, complicated story. It's not going to be a simple story, but one where we have to take many, many things into account. The other, oh dear, all of my layout is going to change. The other thing that we're looking at is that in Down syndrome, it seems that to some degree the brain develops fairly normally for about the first 6 months. And then from 6 months onwards, there's microcephaly, so it gets smaller and smaller in terms of the different volumes. Some new work by a team in London, led by Mary Rutherford, actually shows that even in fetal development, so they're imaging the fetal brain of mothers who are carrying babies with Down syndrome, and they find there are already small differences in volume. So I think the idea that the brain is normal until 6 months is probably going to be questioned very quickly. We are going to be seeing the babies that she is imaging during fetal life after they're born. So we're going to have a very long picture of these developing brains of developing babies. And we're hoping that individual differences, is it not possible to run this off the Macintosh? It's going to be all out of kilter all the time. It's going to overlay. Okay, I'll go on, but I think if I can run it through a Macintosh, it would be helpful.
No, it's okay.
Unless you can change the... She has it on her machine, so if you can, I'll just continue. Anyway, we're going to be looking at the risk and protective factors in babies with Down syndrome. So here are our targets. Our target is to have at least 150 babies from the age of 6 months to 48 months, so when they're still very young in development. We've chosen this because it's a major period of both cognitive change and neural change. The brain changes a great deal during that period, and so does infant cognition. We're going to then take a subgroup of these children and follow them longitudinally. And what we're going to concentrate on are the two extremes, so children who are very impaired and children who are lightly impaired, and then try and form two different subgroups and look to see what the risk and protective factors might be in these subgroups.
We've already recruited 72 volunteers, and we got this because we were very lucky. The Observer newspaper put out a 2-page article on our work, and I had a whole column saying, please contact me if you have a baby with Down syndrome, and I was flooded with emails. So people are very interested now. I think something has really changed, and you've probably sensed this as well. In the past, when a couple had a baby with Down syndrome, they were sort of condemned to having a terrible child. Now parents have really changed and seen what you can do in terms of intervention, etc. And they're much more anxious to be part of research, to do everything they can to bring their child to its own full potential. And that has been a major change in attitude both in parents and in the government. Now I don't know where I am. We recruited also, I don't know again whether this happens in Chile, but we have very many parental groups. So there's one called Downright Excellent, using the word down but also talking about excellence. They're a parental group and they gave us a lot of volunteers. There are other groups like 21 and Company, so Chromosome 21. They're formed by young couples who are really very anxious to have their children part of research and to understand the syndrome in much greater detail than people were prone to in the past. So, we're still, 150 babies is not many when you're doing studies like this. So we're anxious to recruit other groups, and we've been talking to Marcelo and Ricardo about whether or not we could run a similar study here in Chile. We have another collaborator in East London, which is a very poor area of London, because we're interested in differences in socioeconomic status. We have Susan Rivera in Mind Institute in California, who's very interested to collaborate, and Ruth Campos in Madrid, who's already put in a grant to try and do the same protocol as us, so that we have much, much larger numbers. Because nowadays, in the kind of multidisciplinary research that is done, we need much greater numbers than if we're doing a single experimental study. What's interesting, I think, is that all of you who've done experimental work, if you have outliers, you take them out of your analysis. They're a nuisance to the analysis. In this study, we're actually going to focus on outliers because we're interested in the two ends of the continuum, those who are doing very poorly and those who are doing really well. And here's an example of that. So this is one of the tasks we use. And I show it to you now because I want to show you how many differences there are in individuals. So basically, this is the gap overlap task. The baby sees an interesting stimulus on a screen. That stimulus goes away. Something comes up on the right, and they get a reward for moving their eyes to the right. So we were talking over lunch about pencil and paper tasks, etc. When you work with young babies, you have to come up with tasks where you don't give the baby any instruction. You build the instructions, if you like, into the material. So it's, if you get a reward, it's interesting to do that again. And that's how we train babies to do the task. And then we can measure reaction time and so forth. So that's one of the stimuli settings. The other one, the central stimulus stays on when this happens. So here there's a blank screen, and then you get this. But here, the central stimulus stays on. And what we're interested in are attention for strategies such as disengagement. Can the baby disengage from a central stimulus and move their eyes to another one? And the differences between these different tasks will give us measurements of reaction time, of attention shifting, and so forth. And here's just a graph showing you infants with Down syndrome. So this is reaction time, and this is the gap. This is the overlap task. These are the baseline and the ordinary gap task. And what you can see is that for the baseline and the ordinary, all the children look very, very similar. There are no individual differences. But as soon as you get this one where you need disengagement, there are huge individual differences. So some of the children take a very long time to disengage from the central stimulus. And some of them look very much like controls. They're very fast. And it's these children and these that we will concentrate on, and not the ones in the middle. Because the ones in the middle would actually, if we took this as group data, it would get collapsed and we wouldn't see the differences. What we're interested in, as I said, are the outliers. So we're going to get wide individual differences. We know this, all of us, from looking at adults with Down syndrome. Some could do a university degree, some can't even learn to tie their shoelaces. So there are really huge individual differences, and one needs to know why. And so we'll be looking across different cognitive domains, different patterns of neural responses, and I'll show you how we're going to do this in a moment. We're going to link these differences with our genetics colleagues to differences in alleles on the different genes that we're looking at, and cellular differences. So one of our other colleagues is going to be growing stem cells from hair follicles, actually, and then looking at the differences that these give rise to. And these will all be tested by mouse models, because we have another group working with us who have a Down syndrome dementia mouse model, and they'll be testing this. But I wanted to put this other arrow in, because all of the groups is perhaps for me the most interdisciplinary work I've ever done. We often play lip service to interdisciplinary work. We put at the end, and we'll be integrating with so-and-so. But this is really integration, because the geneticists will come back to us and say, wait a minute, we've got these differences here. Can you go back and look at the cognition and see whether there are, again, differences there? Or we'll say to the geneticist, go back and do your analyses again. There must be something different here that's going on. The mouse people will be testing this, and we're integrating with them so that our infancy tasks are very much like the mouse tasks, and I'll show you some of those in a moment on memory. So there's a real feeling of a team coming from very different disciplines, all pulled towards the same goal. So here are some of our methods. I'll go through them all separately in a moment, but I just wanted to point out them. So we're going to be using behavioral tasks with eye tracking. So we have now the Tobii eye tracker, which has nothing on the child at all. We calibrate the eyes from this little camera on the screen, and then we can follow their eye movements over time. We're going to use visual auditory mismatch, so whether the child is sensitive to the integration of what it can hear and what it can see on the mouth. We're going to be using motion capture, which is a system where we put small electrodes on the hands of the child with arms and look at their movements in space, whether they're aiming correctly, how quickly the movement is, and how that changes over time. We're going to look at sleep patterns, and I'll come to that in a moment, but sleep is not only a period of rest, it's a period when the brain is extremely active in some parts of the brain, and other parts close down during sleep. And it means that sleep is a very important indicator for the consolidation of memory. And we know that from many, many studies, but nobody's been working on babies with Down syndrome. We're going to be looking at memory in a lot of detail, because in dementia, memory is one of the things that goes wrong. We're going to be looking at environmental factors. And I'll explain that in more detail in a moment, but all sorts of environmental factors change developmental trajectories, and we need to look at that. And we'll be looking at neural responses using a high-density evoked EEG system. It's a very easy system to put on the baby's head, because while you're playing with the baby with bubbles or something, somebody just slips it on the back of the head, and it measures in 128 different places the electrical activity coming out of the brain. And so we can really get a very good temporal measurement of brain activity. So here's our pilot protocol. We haven't run many babies yet. I think we're on baby 7 now, because it's very intensive. It takes 2 whole days to run with a lot of breaks, obviously. And they're coming from long distances. But we're going to persevere over these 5 years of our grant. So we're going to be taking general measures. So the Mullen scales, I don't know if you use it here, but it's a baby scale. It's a bit like the baby infant scales. But it's an overall test of mental age in emotion, motor, cognitive, and so forth. Yeah. We're going to take mouth swabs to get DNA, and if we find some very interesting DNA differences, we'll then hopefully get blood, but we're not taking blood at the moment, because we can't, at the moment, in our university, take human tissue, and mouth swabs, because they change very quickly and no longer count as human tissue. We're going to take hair follicles as well, so we extract tiny bits of hair, and if we get the follicle, our colleague can grow stem cells from that. We're going to have a lot of parental questionnaires, so poor parents, when they get one of our packages, have hours to fill in, but as I said, they're very motivated, and they do so. As I said, we're going to place a lot of emphasis on sleep patterns, and I'll explain why in a moment. We're looking at parent-child interaction. So we have videos of a few minutes of play with mother or father and child, because again, we think that that changes very subtly when a child is developing atypically. We're going to be taking neural measures, so using this EEG, here in episode that'll go with Down syndrome, we look at resting state brain activity because that's becoming a very important indicator of developmental change. And we're also looking at speech and pitch discrimination because that's a very early marker of language or speech and then language over developmental time. So are children sensitive to differences in changes in vowels from A to U, for instance, or changes in pitch from, I don't know if I can do this, U to U? Yeah. So things like that. And we're looking at various behavioral measures. So the gap overlap task, which I mentioned, will give us a measurement of reaction time, but it will also give us measurements of attention and disinhibition. We're looking at several memory tasks, and I'll go through those in a moment. Motion capture, which I mentioned, but we can look at unimanual movement, bimanual movement, and with toddlers we can also look at gait with this new apparatus. So a lot of things being done over a period of 2 days when the parents come in with their babies. So this is just to show you the motor development. So we get measures from the Mullen on fine and gross motor development, and then we have this motion capture, which will give us very, very detailed information about the development of the motor system in terms of the hands and arms to begin with, and when they're walking in terms of gait. So why do we use eye tracking? Well, first of all, it gives you, I hope you can see this, a very good heat map of where the child is looking. So we get colors that will tell you how long the child looked here and how long it looked in another place. But we also, and above all, get the pattern, the actual tracking pattern. We can see over time where the baby's eyes have moved, not just where it landed. So this is a very important measure, in fact. And we can do this with many different things. So here's one task we have, which is called the face pop-out task, where we have an array of different objects on the screen, one of which is a human face. And what we're interested in is, given these really interesting things, does the baby nonetheless orient more often, come back to the human face? So it may not be the longest thing it looks at, but does it keep coming back? Because we can look at the tracking. This is another one where we're looking at eye gaze. Does the child follow eye gaze? And what is the important thing that the child scans when looking at a face? This is the gap overlap task I told you about. And this is one where we're looking at referential meaning. So when somebody says, look, and turns their face, does the baby follow that and anticipate that that's where something interesting will occur? But here's one reason why these tracking data are so important. So this is another study, but I was looking at number development in babies, sensitivity to numerosity, if you like. And we found very different patterns across 2 different syndromes. So here's a Down syndrome infant and a Williams syndrome infant. We show the infant 8 dots, and the Down syndrome infant clearly goes all around all the dots and even comes back to some. Whereas the Williams infant stays very, very focused on just a few. And we see this again when we look at 16 dots. So the Down syndrome infant doesn't look at them all, but gets a good global overview. The Williams syndrome infant is doing very poorly here. This actually predicts whether or not they're sensitive to changes in small number, so 2 to 3 objects, or changes in magnitude, 8 to 16 objects. The Down syndrome infants do far better than the Williams when it's 8 to 16, so global magnitude. The Williams infants do far better than the Downs when it's small numbers because they concentrate on local objects. So there are interesting implications about what you would train. So in Williams syndrome, you might train them to make a lot of saccadic eye movements. And in the Down syndrome, you train them to stay sustained on individual objects. And those training regimes could have an impact not only on number, but on face processing, on all sorts of other things that involve the movements of the eyes. But what we're interested in, so these are kind of an amalgamation of group data. But what I'm interested in now is within syndrome differences. So imagine that most of our Downs have this global way of looking at an array, but some of them don't. or most of our Williams are very local, but some of them aren't. What does that have in terms of implications for the rest of their cognitive profile? So we're now going to be not grouping our data, but actually looking at individual data. We've got several memory tasks, and we've been piloting, we're almost ready now, but they go from deferred imitation in a live setting to all sorts of computerized tasks. So let me just quickly show you some of them. So this is one of my colleagues. He's at this point working with a typically developing baby. He actually demonstrates a series of steps in a sequence of events, and then stops and then gives the objects to the baby to see whether they copy them. Do they copy all of the sequence, some of the sequence? Is it in order? Is it out of order? And so forth. I'll just show you the pictures from this. It's not, the details are not important. Okay. Paired associate learners. If you watch the screen, not me now, because I'm going to do a lot of things on the screen now. But this is one study where, again, instead of asking people to make a choice or anything like that, we're just looking at where the child looks. So here's a problem for all of you, although I can't make the noises. So you see that on the screen, and it goes boing, 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 boing. And then you see this, and it goes beep, 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 beep. And you see that 6 times, again and again and again. And then all I do is show you the screen blank, because the curtains have come down, and you hear boing, boing, boing. Where are you going to look, left or right? Where do you expect an object to appear? Okay. And what we look at is whether or not when the child hears boing, boing, they look much more on the left. They might move to the right because nothing is happening on the left yet. And when they hear beep, 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 they should look to the critical square on the right-hand side. Now, what's interesting about this in many of these experiments that I'll show you is there are no instructions. We just show the child something again and again so that they learn something, and then we ask them to predict where something may appear. This is a memory for objects task, and from now on, all the tasks are very similar to tasks we do with adults, although the paired associate learning is as well, but also tasks we do with mice. So with our model mice who have Down syndrome and dementia, what is their behavior like in tasks of this kind? So as you can see here, you've got 4 objects, and I want you to watch the screen again. Well, I hope you all noticed the new object. If you haven't, see me afterwards. But basically, what we're doing is in each set of stimuli, we're changing one of the objects to see, and of course it changes place each time, to see whether or not the baby has remembered the 4 objects and noticed a change. Here's another task, which is memory for context, which is an important task. So you see, watch the screen again, please. We're supposed to be learning. Okay. I hope you've all noticed that the background is on a different background. So again, we're trying to get an episodic memory. It's not easy in infants, but this is a change of the object on a background that you've learned to associate with. And we're looking to see whether the baby, I think one thing I didn't say is that infants, if you don't know about infancy research, will always look longer at something they consider novel. So if they've learned something and something new appears, if they've noticed that, they will orient and look longer to that. Okay, why do we use high-density EEG? Well, I'm sure I don't have to convince many of you here, but basically it's because we get, first it's really easy to put on babies. As I said, somebody's playing with a baby with bubbles in front, you just slip it on their back, it's all set up and they go. The second is it really gives you very fine temporal activity of the brain. So you get peaks and valleys, and you can see exactly where they are over time, and you can link them to the stimulus that you're showing in terms of milliseconds, so really important. It also gives you a scalp map. The scalp map isn't as good as fMRI, for instance, but it is a good map. You can really see some quite fundamental differences. You can use it to measure resting state in EEG, and as I said earlier, that's become a very important marker in developmental disorders and in normal development for changes over time. You can link, as I said, to any stimulus. You can link it very, very precisely to the onset of, say, seeing a face. You know exactly the moment at which a face came on the screen, and you know what the brain activity is like. And why it's important is that often scores look very, very similar in developmental disorders to, say, the normal case, but the underlying neural activity is different. I just want to give you an idea of that from some old work of mine using Williams syndrome. They're very, very good at face processing. So if you run standardized tasks with them, they will score in the normal range, and people thought they had an intact face processing module. But with one of my students, we looked at the neural activity underlying face processing, and she compared human faces, monkey faces, and cars. I'm going to go through this quickly because I just want to show you how important it is to actually look at neural processes. So here are the controls. They show a very strong N170, which is about 170 to 200 milliseconds after they see a face on the screen. The brain is saying, this is a face. For both human and monkey faces, but not for cars. The signature for cars looks different. That is not the case in Williams syndrome. First of all, they don't show the strong N170. But second, the whole pattern for cars and faces is very, very similar. So although they're good at face processing, they're not doing it in the same way neurally, and they're certainly not doing it in the same way cognitively. They're much more featural. And we can see that again if we look at upright and upside down faces. So for controls, you see when a face is upright, a strong right hemisphere localization, but when the face is turned upside down, you see bilateral activity. In Williams syndrome, there's no statistical difference between the two. And that suggests that they're very much featural processes when we look at faces. So, although you have scores in the normal range, you have different neural processes and different cognitive processes. Hence the importance of always taking these neural measures if you get scores in the normal range. What about the role of sleep? Well, the expression of that gene that I told you about that is on a chromosome 21 actually differs across the wake and sleep cycle. So it's a very important gene for studies of sleep. The other thing that's been discovered recently, I think I might even have this down here, yes. So changes in sleep in the normal population, elderly population, are about 10 or 20 years prior to any signs of dementia. So when people have done retrospective studies, they found that actually sleep patterns, and that people had gone to the doctor because their sleep patterns had changed radically. And they looked at that in a group with dementia and without at the same age, and they found a real difference. But the other thing is that now it's been recently shown that this accumulation of these amyloid beta plaques is actually affecting not the biological clock itself, it doesn't switch that off, but it interferes with its connection to sleep-wake patterns. So it's actually playing a role in the brain that is interfering with the normal activity of sleep and wakefulness. So we think that this, because the babies have amyloid accumulation much earlier in development, we may see differences in sleep patterns that tell us something about risk and protective factors. I just said that. So the other thing that's important about sleep, and I can't go into the details now, is that it really plays a role in the consolidation of learning. So there are a lot of animal studies, but there are also human studies, and I have a student working on older children with Down's and Williams syndrome that show that sleep plays a very critical role in whatever you have learned the night before. So if you learn something and sleep, you actually wake up with more knowledge than if you learn something, have the same period of time awake and are tested. So sleep is playing a really important role in the consolidation of learning. But we only have group data on this so far. We've used always group analysis. I mean, that's the way we psychologists work. And these individual differences are going to become absolutely critical to the present study. What about environmental? Why environmental? How am I doing my time? Okay? Yeah? So, one thing I want to stress is that you don't just have trisomy 21. You also have the correlation of your IQ with your parents. Of course, your IQ is much lower, different to what it would have been if you hadn't had chromosome 21. But you still have this family resemblance that gives rise to individual differences. We're going to therefore take some parental measures, and we're going to use a very quick go-no-go task to look at attention, basically. And if we find interesting individual differences in the babies, we'll go back then and test the parents in great depth. This will just give us a first quick measure. It takes very little time, and the parents don't feel they're having their IQ tested. They feel it's just a speeding thing, and they quite enjoy doing it. So that is working rather well. So we'll go back and revisit those parents where we have either the very high-functioning or low-functioning children. But also, we know from other studies that I've been doing recently that SES plays a great important role in brain structure. So I just want to show you this because we'll be looking at this with the Down syndrome population. So we had a group of children from a very poor part of London who are very low in socioeconomic status, and we compared them to a group who had much higher socioeconomic status. And we were looking at resting state brain activity. And we looked at the gamma band, which is really sort of to do with integrating information. Is the quickest way to explain it. And what we found was that in most areas of the brain, temporal and occipital, etc., there were no differences between the infants. But in the frontal part of the brain, which is the area that's involved in monitoring, planning, forward-looking, and so forth, already by 6 months, there was a difference in brain activity. And that was a function of SES, which is very frightening, actually, but very real. And the results were staggeringly significant. We've published that if anyone would like to look at that in more depth. So this was the first study that showed region-selective deficits caused by SES. All other things were accounted for. And as I said, as early as 6 months. So 6 months into life, the brain is already changing in its activity as affected by low SES. So we'll be measuring SES as one of the factors that I haven't measured in the past very much, because I'm not focused on in our Down syndrome population. We'll also look at parent-child interaction, because other work that we did with typically developing infants some years ago showed that the style of interaction, whether the mother is more I'll say mother, but I mean parent in general, more controlling or more sensitive, affected the milestones at which, the age at which babies reach certain milestones. Things like speech discrimination, understanding causal events, all of this in infancy, so with these very simple tasks, were affected not across the board. So sensitive mothers' babies were earlier to reach discrimination of speech, but later to reach causality. More controlling mothers were earlier to reach causality and later for speech. And we think it's due to differences like if you're a very sensitive mother, you're always up in the ante slightly with the baby's productions. The baby says ah, and you repeat ah, and then you go ma to add to it and so forth. Whereas the more controlling mother speaks directly, we're all laughing looking at you, speaks more directly to their child and doesn't adapt their language. However, if you're looking at causality, if your mother is always adapting to you, you don't have to work out what the mother's goals are. She's always working out what your goals are. Whereas if you're a more controlling mother, the baby actually has to work out what its mother's goals are and therefore gets causality early. Why I mention this is because obviously the effect of mother-child interaction is not only in typical development, it's also in atypical development. So we should see some differences there. So we're taking these measures of mother-child interaction and analysing them in terms of how directive they are and so forth. And we know, for instance, from work that we've done with Down syndrome, that this really has an effect. And what I'm asking is, why does a healthy, so most of the children who we look at, who come in as volunteers, have a really positive, healthy, stimulating environment. They're not from low SES families. They will be in future because we're recruiting like that. Most of them so far come in are not. And yet that doesn't compensate for the genetic vulnerability. So why is this? Is it just, as many people think, just because of the genes? What we wanted to show was it's not just genetic. The moment a parent is told your child has Down syndrome or Fragile X syndrome or whatever, the parental attitude changes, expectations change, the way they correct their child changes. So there have been a couple of studies. One is from my own lab. It's not really a study, this is more observational stuff, where we looked at just, did the mother let the child mouth objects? When I want to understand what the subject is like and I'm a 6-month-old, I put it in my mouth and I feel its shape, its texture and everything. Mothers of babies with the developmental disorders took them out of the mouth immediately. Understandably, they're much more frightened about danger. Mothers of typically developing infants, as long as it wasn't too small and it was clean, they let them explore. The same with crawling. If they put the baby down and the baby crawled away, the mother of an atypically developing baby, so with Down syndrome, would run and pick the baby up straight away, scared naturally that there would be danger. The mother of a typically developing baby just watched from afar. So in both cases, we're stopping the baby exploring the world by a natural, and I would do the same, I'm sure, fear of danger that is greater when you have a child who's vulnerable. The second example is from Carolyn Mervis's lab, and this is one I think you've also noticed, which is about language. So in typical development, the child goes cat, and it's really a dog, or mispronounces cat and goes cash or something, and the mother says, yeah, it's like a cat, and etc. In the atypically developing child, the child says cat. If it's a dog, the mother says no, dog. And the natural fear is that they won't ever learn. Because they have learning difficulties, you have this built-in fear that you're unaware of, that they may not learn, so you're always correcting. But there's a huge difference between these two. If you let the child call everything a cat, then actually they're forming a little category of animal, which they'll break down later to dog, cat, and cow. But if you stop that, you get this very local one-to-one mapping and no general categorical development. And we know that people with infants with developmental disorders have a lot of problems with categories. So inadvertently, because the parents are being very protective, they're actually stopping exploration and development of categories. So these are just two examples why the environment matters a great deal and why we want to look at it in more detail. So I think we need a much more dynamic view of how the social environment interacts back and forth with the genetic environment of these children. Okay, we have a lot of challenges, and I'm going to end on that. Time is correct. It's a 5-year grant. It's called a strategic grant, and I think the Wellcome Trust did that because they know we may not pull it off, but we're certainly going to try. We want to inform the developmental trajectory of Down syndrome itself, so we want far more detail about the fine details of things like memory and attention and so forth over developmental time. We want to identify risk and protective factors for Alzheimer's disease, and that's underpinning the whole grant, but there'll be a lot of other things that come out of it. We want to really try and explain individual differences, and it won't be one factor that explains why some are better than others. It's going to be a very complex interaction between multiple factors, and I think that's something of a real challenge, and it's a challenge statistically as well, how to actually take these things into account, and we've got a very good statistician on board to help us with that. But we also want from all of this to suggest early intervention strategies. So not to intervene at, say, age 5 when the child is showing already that they can't read or they can't have a good sensitivity to number, but to get in much earlier on much lower level processes. So for instance, we would train on attention, sustained attention, movements of attention, attending to subtle differences and so forth. So very low level basic processes trained really early in development, which would then, we hope, cascade over time on the developing cognitive level domains. So we'd avoid cognitive level training for basic process level training. And we'd also look at mother-child interaction in very more detail than we've ever done in the past, and try and set up training studies that would change this more directive style, the understandably directive style, to a more subtle one. When I think parents need to understand why it's important to make errors. I think that's something that we often forget. You know, because if our child is developing typically and they make errors, we don't think anything of it. We know they're going to get there in the long run. But if you're the parent of an atypically developing child, you worry about things like errors. Well, actually, errors are incredibly important for learning. And so we want to train parents to think differently about the way their child is developing. And with that, I'd just like to recognize, so this is the Lung Downs Consortium. Andre Stridham is the person working on adults with Down syndrome and those with or without dementia. John Hardy is our geneticist in charge of the genetics groups. Liz Fisher and Viktor Turkovic are the mouse model people. Dean Nichevich is the person who grows stem cells. He's quite brilliant, quite ununderstandable. And that's me. And my little team who work on the infant cohort, Isha Marchand is the postdoc, George Ball is the research assistant, and these are PhD students. Thank you very much. So I'm happy to answer any questions. You can say it in Spanish if you like, but I might need some help understanding.
My question is just methodological regarding the resting state EEG you were showing. Why gamma first, and how do you record this in resting state? Because I mean, much of the activity of the resting state is very low frequency, and EEG is not necessarily the best for very low frequencies.
I
So
agree. We call it resting state because everyone else does, but there's actually a stimulus that is there. We don't only look at gamma. I reported gamma because in fact that was the one that showed the differences in the SES study, but we're also looking at the other frequencies as well.
sorry, you call it resting state in what sense then? It's just they're looking at something. It's
yes. There's no task, so they don't actually have to do anything like anticipate where something is coming. But a lot of people are worried about this, but it's the same in fMRI when they talk about resting state. It's a misnomer. I don't think anyone's come up with a better expression. But the point is there is no task. So we're looking at in-between task activity when they're just looking at something happening on the screen. But you're right to raise it. I should say there's never a stupid question. Whenever one asks a question, it makes the speaker think and think, oh, I wish I'd thought of that before. So good questions always.
Hi, I have a question with the reaction time.
Yeah.
Yeah, I'm wondering how you get the baseline that was very close to the gap condition.
Oh, okay. The baseline condition is one where there is no gap, so the thing comes on immediately. And so we just look at how long does it take the child to move their eyes. There's no clue in between. And so we then subtract gap from baseline and
An overlap?
then overlap from gap. Yes, thank you.
Okay, thanks.
But it's a nice measure of reaction time. And now we have a very good man on our overall team at the Center for Brain and Cognitive Development called Tim Smith, and he's come in from perception psychology, but he's really interested in scene analysis, the visual scene analysis. And he suggested to us the other day, and I think it's a really good suggestion, that we take the eye tracking data from all our tasks ignore what the task is and get some general measure. And we can, of course, with reaction time, with sustained attention, we can take like 4 or 5 measures and we can get very much power because we'll group all the different tasks. So we'll be analyzing it task by task, but we'll also do this other thing, which I think is really nice in baby work, because otherwise, and there's another thing too, movements of the eyes seem less, I'm not going to say totally, influenced by motor development, if you like. So when you look at reaction time, you've got a motor response. It's very hard to separate the two. You can do it with inspection time. So you can have things on a screen and you look to see how long does it take a person to make a decision before the stimulus disappears. And you can separate out that from how long does it take them to press the button by using a step function. With babies, it's a little bit more tricky, but this kind of gets it.
I was fascinated by the differences between the parenting of parents with Down syndrome and typical development. I was not aware of that. And I think it's a very good hypothesis that maybe, and I share it, that maybe the mothers or the fathers think that, you know, they have this, oh, they will always call the cat the dog, and so they correct them. But could there also be some interaction between the child's behavior? So maybe the children, it's hard to determine where the differences start, right?
Sure.
But do you have any evidence as to whether it's the child's behavior that's driving it, or how would you differentiate between these two hypotheses, that it is the parents' belief that's driving it?
I think there are 3 hypotheses actually. One is it's driven by the parents and their beliefs about the child will never learn. The other is, as you suggest, it's driven by the child because they're not learning, and so it becomes something that the parents are sensitive to. The third is it's a very complex interaction with the two, and I think that's going to be the right one. This is particularly true in the case of autism. So if we look at autistic babies, they don't react in the same way as, say, a Williams syndrome baby will always smile if you smile at it. The autistic baby doesn't. Who's driving that? I don't think it's one or the other. It's very, very complex interaction. Interactions are really hard to measure.
Do you have any ideas of how to look at that?
Yeah, I don't. I think one thing I would want to do is look developmentally. So when you know, so in some cases, like Williams syndrome, you really don't know until the baby's a few months old that it has Williams syndrome. They start to show heart defects, etc. But with Down syndrome and with baby siblings of children who are at high risk of autism, dyslexia, ADHD, etc., you could look really from the start. So you really need to get in the moment the child is born and look at some of those very complex interactions to see what's driving what. But my guess is it will always be an interaction.
Yes.
And how to measure that, I think, is a real challenge for the field now. So, and it's true not only in terms of human interaction, but the interaction of genes with behavior, because they're all double-way arrows. It isn't gene that makes the brain in such a way. The brain develops and changes gene expression. We know that from many studies now. It's tough. It used to be easier, but it's not. I wish there were one-to-one mappings sometimes, but then I think, no, that's not the way human development. It's not the way biological development is. Everywhere we look now, it's complex. So I'm going to call an article. We were talking at lunch about writing theoretical articles, and I'm going to call one Embrace Complexity. Because people often put their hand up and say, wouldn't it be simpler to do X? And I say, yes, but we've missed the point. So we have to embrace complexity, however hard that is. And we need very brilliant statisticians to help us work out how to analyze the data, because it's not obvious in the way that perhaps it was in the past.
Regarding when you talk about the different patterns of eye movement in Williams and in Down syndrome, and you said something that sort of lifted the flag when you said we want to train them to look a different way, and I was wondering what evidence do you have that such a training has any plausible effect? I mean, because just wouldn't if the eye movement, one could say this is just the outcome of a certain developmental trajectory. Why by modifying the eye movement? Again, I wish it could be. It would be great, but what do you have as a?
We don't know yet. I think it's a very basic process that you see from extremely early on in development, and if it's going wrong, it's going to compound over developmental time. That's the point of this notion of cascade. So I think it's a good starting point. We may find that we need to go lower than that, but then it's tough. So that is something you can measure from birth, eye movements. So why not attack something like that? There are very few other things that are there from the very start. You can get neural activity from auditory information, of course. But it seems to me like that's something malleable, something we could do with rewarding. So I was thinking about using little lights that change across the screen to entrain eye movements. And then for doing the whole thing, you get some sort of reward. And maybe from that, but whether that has an impact on, say, scanning static numerical displays, we don't know. That's something we have to test. But the same with mother-child interaction. We don't know whether that's going to work. People are doing it in London at the moment with the siblings of children with autism, and training mothers to interact differently. And in 3 years' time, we'll know whether those children who are at risk have gone back to the normal trajectory or not.
And when you talk about attentional training, specifically what type of training are you? I mean, this is one thing with eye tracking, but you talked about sort of
sustained attention.
How do you train that?
We don't know yet. That's what we're sitting and trying to brainstorming about. I don't think it's obvious. It's kind of more obvious when you get older, but when you're a baby, it's not as obvious. But it'll be indirect, and it has to be challenging, fun, and etc. Yeah. But I think also one of the things that we want to do is not only get a change in behavior, but a change in the neural activity that sustains the behavior. So we will be measuring that as well before and after the training. But training hasn't been part of my psychology up to now, so I'm on a fast learning curve to understand how you can do it, what you should measure, and what kind of things you can train easily, and whether they impact. See, I'm not interested in teaching a number. I'm interested in something that will have a wider impact across the cognitive system.
I was wondering if you have any hypotheses about why good environments doesn't help for genetics.
Well, I think it does to some extent, but it doesn't do away with it. So, it's relevant to the question that you were asking, because if you do have, say you have a mother, and you do have some now, with a very, very good sensitive attitude to her child with Down syndrome, that should have an impact, and you should be able to measure that. When we look at these extremes of the two ends of the continuum, we're expecting to find not only genetic differences on other chromosomes than 21, but also parent-child differences and so on and so forth. And we're hoping that those will together explain the extremes. So I think why I was asking is, people assume that if you have a bad environment, you get bad outcome. So why wouldn't you just assume if you have a good environment, you get good outcome? The genetic constraints are there. You can't deny them, but it may be that we can overcome them by certain strategies like good parenting or cognitive stimulation or better diet. I mean, there are lots of factors. Better sleep. I'm convinced that sleep will be a very important factor, and that we can intervene on sleep, of course. And we know children with Down syndrome have apnea, so they wake a lot. They're very fragmented sleep. And fragmented sleep is a predictor for poor cognitive outcome. So I don't think one thing will solve anything. I think it's going to be a conglomeration of things that interact together to get a better outcome. Yeah.
I don't know if I missed it, but how do you plan to connect this with, or draw any conclusions towards the adult dementia part? How do you plan to connect?
Okay.
Yeah, this.
So this is one of the. I think I understood what she said. So she was asking how I connect all this to
Yeah.
the adult dementia. So the reasoning is as follows from Lon Down's thing. Would you move your head slightly, then I can see her. The adult group will be looking at adults with Down syndrome who either have or don't have dementia. And they'll be measuring all sorts of very similar things to us. So if we get outliers, we'll send the information to them. If they get outliers, they'll send it to us. When they send to us, we'll immediately look for those things and send the genetics, alert the genetics people that already have genome-wide association studies done. So it'll be going back and forth all the time. And what we're hoping is that we'll find some very early signs that would indicate a risk factor or a protective factor. Not that it will eradicate or not, which might lead to later onset. One thing I forgot to mention is that in Down syndrome, the onset of dementia is much earlier. It's at about 30 years of age rather than 60 in the typical population. So we might be able to push that back, or we might be able to even avert it. Since we have a 75-year-old with Down syndrome who's completely alert, no cognitive decline. He has Down syndrome, so he has some of the problems of Down syndrome, but nothing has got worse with aging, whereas others at 30 show very clear signs of dementia.
That's why you chose the population of Down syndrome baby.
Yes, so the choice was exactly because this APP gene on chromosome 21 is overexpressed from conception on. So we know that there is a risk, and yet not everyone gets it. You know, I think the really interesting thing is if you look at the brains of people with Down syndrome at about 35, 40, they look like dementia patients. But 40% of them don't have dementia. Why? So that's why it's a very interesting group to look at, because you're sure of a risk factor, and yet not everyone goes on. So the thing that has already been shown is that the APP gene alone does not cause dementia. It causes plaques, but plaques alone don't cause dementia. I'm sorry, I should have been clearer about that in the beginning. Thank you all for your questions. Thank you.
Nos vemos la próxima semana.